Welcome to the video lecture for Module 1.2.4. Today we're talking about the pancreas. As always, here are the learning objectives that I use to write both the quizzes and the exams. All right, so let's jump into it. The pancreas is both an exocrine organ and an endocrine gland, and we'll talk more about that as we get through this. So the pancreas is mostly retroperitoneal, and it lies in the loop between the inferior border of the stomach and the proximal portion of the small intestines. It's slender, pale organ with a nodular or lumpy texture. The pancreas is about 20 to 25 centimeters long, and it weighs about 80 grams in adults. So we'll consider its anatomy further in the digestive units because it's primarily an exocrine organ that makes digestive enzymes. The exocrine pancreas consists of clusters of gland cells called pancreatic acini and their attached ducts. The exocrine pancreas takes up about roughly 99% of the pancreatic volume, and together the gland and the ducts secrete large quantities of alkaline, enzyme-rich fluid that reaches the lumen of the digestive tract through a network of secretory ducts. So the endocrine pancreas consists of a small group of cells scattered among the exocrine cells. The endocrine clusters are known as pancreatic islets, or the islets of Langerhans. Pancreatic islets account for only about 1% of all cells in the pancreas. Nevertheless, a typical pancreas contains approximately 2 million pancreatic islets, and their secretions are vital to our survival. The pancreatic islets are surrounded by an extensive fenestrated capillary network that carries pancreatic hormones into the bloodstream. Each islet contains 4 types of cells. 1. Alpha cells that produce the hormone glucagon. Glucagon raises blood glucose levels by increasing the rates of glycogen breakdown and glucose released by the liver. 2. Beta cells produce the hormone insulin. Insulin lowers blood glucose levels by increasing the rate of glucose uptake and utilization by most body cells, and by increasing glycogen synthesis in skeletal muscles in the liver. Beta cells also secrete amylin, a peptide hormone whose level rises after a meal. 3. Delta cells produce a peptide hormone identical to growth hormone inhibiting hormone, or GHIH, a hypothalamic regulatory hormone. GHIH suppresses the release of glucagon and insulin by other islet cells and slows the rate of food absorption and enzyme secretion along the digestive tract. And 4, F cells produce the hormone pancreatic polypeptide, or PP. PP inhibits gallbladder contraction and regulates the production of some pancreatic enzymes. It may also help control the rate of nutrient absorption by the digestive tract. We'll focus on insulin and glucagon, which are the hormones that regulate blood glucose levels later on. But here's a tip. To help in differentiating between the polysaccharide storage molecule glycogen and the pancreatic hormone glucagon, remember that glycogen literally means generates sugar, and associate glucagon with the pentagon, both of which issue orders. So when blood glucose levels increase, beta cells secrete insulin, which then stimulates the transport of glucose across plasma membranes and into target cells. When blood glucose levels decrease, alpha cells secrete glucagon, which stimulates glycogen breakdown and glucose release by the liver. Insulin is a peptide hormone released by beta cells when blood glucose concentrations exceed normal levels, so about 70 to 110 milligrams per deciliter. Elevated levels of some amino acids, including arginine and leucine, also stimulate secretion of insulin. This hormone affects cellular metabolism in a series of steps that begins when insulin binds to receptor proteins on the plasma membrane of a target cell. Binding activates the receptor, which functions as a kinase, attaching phosphate groups to intracellular enzymes. These enzymes then produce primary and secondary effects into the cell. The biochemical details of these effects remain pretty unresolved. One of the most important effects is the enhancement of glucose absorption and utilization. Insulin receptors are present in most plasma membranes, and cells that have them are called insulin dependent. However, cells in the brain and kidneys, cells in the lining of the digestive tract, and red blood cells lack insulin receptors. These cells are called insulin independent because they can absorb and utilize glucose without insulin stimulation. The effects of insulin on its target cells include the following. So accelerating glucose uptake, which is all cells, and this effect results from an increase in the number of glucose transport proteins in the plasma membrane. These proteins move glucose into the cell by facilitated diffusion, which follows the concentration gradient for glucose and does not require ATP. So accelerating glucose utilization, which has all target cells as well, and enhanced ATP production. This effect occurs for two reasons. One, the rate of glucose use is proportional to its availability, so when more glucose enters a cell, more is used. And second messengers activate a key enzyme involved in the initial steps of glycolysis. Another is stimulating glycogen formation, the skeletal muscles and liver cells. When excess glucose enters these cells, it's stored as glycogen. Another is stimulating amino acid absorption and protein synthesis. This helps to maintain glucose levels by preventing the conversion of amino acids to glucose. Another is stimulating triglyceride formation in adipose tissue. Insulin stimulates the absorption of fatty acids and glycerol by adipocytes, which store these components as triglycerides. Adipocytes also increase their absorption of glucose, and excess glucose is used in the synthesis of additional triglycerides. So, to summarize, the pancreas secretes insulin when glucose is abundant, and the hormone stimulates glucose utilization to support growth and to build carbohydrate or glycogen and lipid triglyceride reserves. The accelerated use of glucose soon brings circulating glucose levels within normal limits. A key point to remember is that the function of insulin is to get glucose into cells. When glucose concentrations decrease below normal, alpha cells release glucagon to mobilize energy reserves. When glucagon binds to a receptor in the target cell's plasma membrane, the hormone activates adenylate cyclase. As we've seen, CAMP acts as a second messenger that activates cytoplasmic enzymes. The primary effects of glucagon are as follows. One, it stimulates the breakdown of glycogen in skeletal muscles and liver cells. The glucose molecules released are either metabolized for energy in skeletal muscle fibers or released into the bloodstream by liver cells. Two, it stimulates the breakdown of triglycerides in adipose tissue. The adipocytes then release the fatty acids into the bloodstream for use by other tissues. Three, stimulates the production and release of glucose by the liver. Liver cells can absorb amino acids from the bloodstream and convert them to glucose, and release the glucose into circulation. This process of glucose synthesis in the liver is called gluconeogenesis. The results are a reduction in glucose use and the release of more glucose into the bloodstream. Blood glucose concentrations soon increase towards normal levels. Pancreatic alpha cells and beta cells monitor blood glucose concentrations, and they secrete glucagon and insulin without endocrine or nervous instructions. Yet, because the alpha cells and beta cells are highly sensitive to changes in blood glucose levels, any hormone that affects blood glucose concentrations indirectly affects the production of both insulin and glucagon. Autonomic activity also influences insulin production. Parasympathetic stimulation enhances insulin release, and sympathetic stimulation inhibits it. So you have a lot of information about insulin and glucagon and other pancreatic hormones that are summarized in this table 18-5 that you see here. I do want to take a little bit of time and highlight one of the things that's probably on everybody's mind whenever we talk about glucose and insulin, and that is diabetes mellitus. So the figure here is a little hard to see. I included blown up portions of the text on the next few slides and some breakdowns that you can see there that go over it. I'm not going to go into diabetes too much as far as the video is concerned, because most of you can read all of this information here. And most of us, at least at this point in our level of academia and anatomy and physiology, probably have a good idea of what diabetes is. But what I will say is that whether glucose is absorbed by the digestive tract or manufactured and released by the liver, very little glucose leaves the body once it's entered the bloodstream. So the kidneys reabsorb virtually all glucose. So glucose does not appear in the urine. However, in diabetes mellitus, glucose accumulates in the blood and urine as a result of faulty glucose metabolism. Diabetes mellitus can be caused by genetic abnormalities, and some of the genes responsible have been identified. Mutations that result in inadequate insulin production, or the synthesis of abnormal insulin molecules, or the production of defective receptor proteins to produce comparable symptoms. Under these conditions, obesity accelerates in the onset and severity of the disease. Diabetes mellitus can also result from other pathological conditions, so injury, autoimmune disorders or immune disorders, or hormonal imbalances. And I'm sure everybody knows about these, so we won't delve too deeply into them, but just know or make sure that you read over both type 1 and type 2. So we have two major types of diabetes mellitus, and they are type 1 diabetes, or the insulin-dependent diabetes. And we also have type 2 diabetes, used to be known as adult onset diabetes, but as we progress to a more unhealthy lifestyle, especially here in America, we're starting to see more children be affected by type 2 diabetes, where originally we would only see children affected with type 1. So, in addition to the rest of these slides that you see here, and you can go through and read that on your own, I want you to know some things and keep some things in mind. I want you to be able to identify the types of cells in the pancreatic islets and the hormones that are produced by each. And I want you to be able to answer a few questions as well. Why does a person with type 1 or type 2 diabetes urinate frequently and have increased thirst? And what effect would increased levels of glucagon have on the amount of glycogen that's stored in the liver?